Hello, we are Group 10 and we are currently on a farm where Chlamydia abortus is the suspected cause of abortion among sheep. We will further discuss the various aspects of this disease.
Hello, my name is Lorato. Firstly, I'll be defining what Chlamydia abortus is, and later I'll be telling you about the lab test performed to diagnose this disease in bovines. Chlamydia abortus is classified as a gram-negative intracellular bacterium and a zoonotic agent.
Hi, my name is Vaishnavi. I will be discussing the characteristics of Chlamydia abortus and whether or not it can survive outside its host cell.
Characteristic features of Chlamydia abortus. This bacteria has two membranes, no flagella, which means it is non-motile. It is a gram-negative obligate intracellular bacteria. It is pleomorphic and has a unique life cycle inside its host in two different forms, the non-infective reticular body and the infective elementary body. Can Chlamydia abortus survive outside a host cell? Because Chlamydia abortus is a bacteria which is a living organism, it can therefore survive outside its host.
Hello, my name is Kayla and today I'm going to talk about this recommended samples to be taken and how to preserve and transport these samples.
Given the characteristics of the causative organism, which types of samples would you take to send to the laboratory? Some animals may shed Chlamydia abortus in vaginal fluids for more than 2 weeks before abortion, up until 2 weeks after abortion. A sample for testing can be taken from these fluids. Samples can be taken from the aborted fetus. The placenta can also be used. Vaginal swabs may be taken for laboratory testing. The still moist fleece of recently aborted fetuses can also be used. Blood samples taken from the sheep following abortion. How will you preserve and transport these samples? Small fetuses should be submitted intact along with the placenta. They should not be put in formalin. Send fresh, chilled and overnight. Transport vaginal swabs in a sterile, dry-capped tube. Send blood samples in a closed container.
Diagnostic techniques include identifying the agent and performing a serological test. The agent can be identified, number one, using smears that can be made from the chronicle filly or chorion on the planta. If the placenta isn't available, vaginal swabs can be taken. Staining procedures that help the smear to be viewed under a microscope include Machiavel, Gisma, and Zeal-Nielsen stains. Number two, antigens detections. Chlamydia abortus antigens are mixed with the sample swabs from the uterus. If chlamydia is present in the female, there will be antibodies specific to the antigen in the sample. This will form clumping which can be viewed as positive test for Chlamydia abortus. Number three, the tissue section Gisma staining can be used to study the presence of Chlamydia abortus in the thin tissue samples taken from the target tissue. Isolating the age. Number four, Chlamydia abortus can be isolated in the embryonate chicken eggs and cell cultures. Chicken eggs are inoculated and then incubated. The affected embryos die within 4 to 13 hours of inoculation. The cells cultures can be prepared in buffalo green monkey or baby hamster kidney. The cell cultures are then incubated and later studied using the Gisma stain. Serological tests can be performed using number one, complement fixation test. Paired sera and are collected one at a time of the abortion and 3 weeks later the other. If the sera shows a rise, complete fixation, antibiotic tired, a conclusion can be made that the animal is carrying Chlamydia abortus. This is the most widely used procedure used for detecting the infection. Number two, microimmunofluorescence test. This procedure is normally 2 time consuming for routine diagnosis purpose. Number three, ELISA. This can be used to detect the infection but doesn't show the species specificity.
My name is Helen Aldrich, and I'll be covering how this disease or organism is transmitted from one animal to the next. Chlamydia abortus is found in the placenta of aborted lambs as well as in the vaginal discharge from the ewe. Transmission can therefore occur in the following ways. Firstly, it can occur orally through the ingestion of fetal membranes of the aborted lamb by the dam or other ewes, or the licking of sniffing by other sheep due to curiosity. This can cause the transmission of Chlamydia abortus. Secondly is the aerosolic method, in which microbes are displaced into the air upon expulsion of the fetus at birth. Thirdly is the sexual method, in which if a ram attempts to mate with an infected ewe, he could acquire the disease via the urogenital tract. The final method is any physical contact with the bacteria. For example, if any uterine fluid that is excreted by the ewe comes into contact with other animals. Another example of transmission that takes advantage of the natural behaviors of the ewe is 2 to 3 days before or on the day of ovulation. Ewes will always shed more vaginal fluid whether healthy or infected, and if this fluid contains the pathogen, it can be a very effective route of transmission to other ewes or healthy rams. Chlamydia abortus infection can be clearly observed by the presence of placentitis or infected placenta, which manifests in a red and bloody placenta among the fetal membranes at birth.
My name is Liane, and I will be telling you about the life cycle of the family Chlamydiaceae. The family Chlamydiaceae has a very interesting biphasic developmental cycle. As you can see in the top picture, it starts with an elementary body, the infectious, metabolically inactive bacterial form, entering a eukaryotic cell by endocytosis. Whilst inside this cytoplasmic inclusion, the elementary body then transforms into a reticulate body. This is the metabolically active, non-infectious form of the bacteria. The reticulate body replicates through binary fission. Once the inclusion is filled with its progeny, some of the chlamydiae transform back into elementary bodies. When the host cell ruptures, or if the inclusion fuses with the plasma membrane, they are released to begin the cycle anew.
Hello, I'm Garrett Gregory, and I will be talking about the difference between Chlamydia abortus and other bacteria. How does Chlamydia abortus differ from other bacteria? In terms of classification, Chlamydia abortus belongs to the domain Bacteria, the order Chlamydiales, family Chlamydiaceae, and genus Chlamydia. Morphological characteristics. Chlamydia abortus is roughly spherical, measuring between 0.3 and 1 micrometers in diameter, according to the stage of development it is in, either elementary body or reticulate body. Other bacteria can occur as either rods, cocci, helical forms, or occasionally branching filaments, ranging from 0.5 to 5 micrometers in length. Chlamydia abortus has complete cell walls, similar to gram-negative bacteria, and beneath the cell wall lies a separate cytoplasmic membrane comprising of large amounts of lipid. Other bacteria have cell walls more similar to those of gram-positive bacteria. Chlamydia abortus does not have a nuclear membrane. Chlamydia abortus cells does not have a capsule or flagella. Other bacteria has capsules and flagella. Reproduction. Replication occurs through binary fusion, as happens with most bacteria. Laboratory identification techniques. With direct microscopy, as it is suitable for the detection of organisms in smears or staining procedures. With serological procedures for the detection of antibodies to chlamydia, through culturing, molecular techniques like PCR, real-time PCR, and microarray assay.
Hello, I'm Kayla, and today I will talk about the differences between chlamydia and viruses.
How Chlamydia abortus differs from viruses. Viruses, not considered a living entity. They are very small, can only be seen with an electron microscope, acellular, and they contain only one type of nucleic acid, DNA or RNA. Chlamydia is considered a living entity. They are bigger than viruses, relatively simple unicellular organisms, and they contain DNA, RNA, and ribosomes. Morphological characteristics. Viruses are classified according to their capsid structure, which can be helical, polyhedral, enveloped, or complex. Chlamydia cells is roughly spherical. Reproduction. Virus. For a virus to multiply, it must invade a host cell and direct the host's metabolic machinery to produce vital enzymes and components. Viral multiplication can be demonstrated by a viral one-step growth curve. Chlamydia. Unique biphasic developmental cycle characterized by two distinct morphological forms, reticulate body and an elementary body. Replication by binary fission, typical bacterial growth curve. Laboratory identification. Virus cannot be seen without the use of an electron microscope. Serological methods like Western blotting are the most commonly used means of identification. Can also be identified by using RFLP and PCR. Chlamydia. Whereas chlamydial cells can be identified by direct microscopy and also other methods previously discussed.
Oh my god, does my hair look nice? With that fun guy you were telling me about?
No. Fungi. Hi, I'm Yeshni Govender, and I'm going to be telling you about fungi and how they are compared to Chlamydia abortus.
Fungi versus Chlamydia abortus. The classification of fungi. Fungi belong to Eukaryota. Yeasts, molds, and mushrooms are classified as fungi. Fungi are classified as non-photosynthetic eukaryotes. The kingdom or domain fungi contain 5 major phyla: Chytridiomycota, Zygomycota, Ascomycota, Basidiomycota, and the recently described phylum Glomeromycota. The Deuteromycota is an informal group of unrelated fungi that all share a common characteristic in that they strictly use asexual reproduction. Well, what do fungi look like? Fungi may be either unicellular, the yeasts, or multicellular. Multicellular fungi produce filamentous structures called molds. In molds, the cells are cylindrical and attached end to end, forming branched hyphae. The cell wall of fungi contain chitin and other polysaccharides. Yeasts have an oval or spherical appearance. Septa or cross walls are often found in hyphae. Reproduction. Reproduction in fungi is done by spore formation, which can either be asexual or sexual. The former fungal species during its sexually reproductive life cycle is termed its telomorph, while the asexual form is termed its anamorph. The preferred term for fungi that lack a meiotic stage is mitosporic fungi. During asexual reproduction, 2 spores are produced, conidia and sporangiospores. In most yeasts, asexual division is by budding, and some use binary fission. Fungi can also reproduce by fragmentation. What lab techniques can you use to identify fungi? Most fungal species are identified according to their morphology and sexual reproduction. Molecular methods can be used to classify fungal species. The grouping is based on nucleotide sequence of high conserved ribosomal RNA. Identification of pathogenic fungi is done through direct microscopic examination of wet preparations using morphology.